Okay, chapter 2, plant cell. Okay, what is cytology?
Okay, cytology is the branch of biology concerned with the structure and function of plant and animal cell.
Mm-hmm.
Okay, cell is the basic unit of life.
Okay.
The first is cell, tissue, organ, system, and last organism. What is the important term of plant cell?
Okay, the important term for the plant cell is first protoplasm, the colorless material comprising the living part of a cell, including the cytoplasm, nucleus, and other organelles. Second, protoplasm is plant cell without cell wall. Third one, cytoplasm, material within the living cell, excluding the nucleus. And fourth one, cytosol, aqueous portion of cytoplasm contain water and dissolved substance. For example, amino acid, protein, enzyme. Fifth one is nucleus plasm, liquid and organelles in the nucleus. Okay, six is organelles, that is organized or specialized structures within a living cell. Okay, number seven, prokaryotic cell, single cells organism with neither a distinct nucleus, which are membranes, nor other specialized structures. Okay, for at number 8 is eukaryotic cells, organism consisting of a cell or cells in which the genetic material is DNA in the form of chromosomes contained within a distinct nucleus. Okay, what is cell structure?
Cell structure is the cytoplasmic membrane and cell wall.
Mm-hmm.
Each have a nucleus and organelle. The cytoplasmic membrane and cell wall, aka plasma membrane or cell membrane, that each have a thin structure that completely surround the cell. Typically 7.5 and to 8 micrometer thick. Structure fluid mosaic model. The function is to support and protect cell function, then defend against pathogen and provide shape to the plant cell. What is the function of cell membrane?
Okay, the function of cell membranes is regulating the movement of materials into and exiting the cell. For example, highly selective, selective permeability, and then give shapes, strength, and protection to cell.
Can you describe about nucleus?
Okay, the genetic material of eukaryotic cell is found mainly in nucleus. Okay, most important organelles and twinkle 5 micrometer in diameter. Okay, structure contain nuclear membrane. For example, two layer, inner and outer layer. Outer layer is continuous and connected with another organelles, the rough endoplasmic reticulum. Chromatin is contain DNA and proteins. Nucleolus is a region in which RNA are synthesized and aggregate for nu- nu- nuclear for 100 micrometer in diameter, allowing RNA and protein to move in and out from nucleus. Okay, what is the function of nucleus?
Okay, the function of nucleus is regulating cell activities which are continuously happening by determining which protein molecule should be produced. Then it's involved in heredity function and involved in cell division. The location of ribosome synthesis, for example, nucleolus.
Okay, can you describe about the plastid?
Okay, plastid only exists in plant cell. There are three types. First is chloroplast. Green, it's a green colored plastid, which is chlorophyll. Second is chromoplast. The color is orange or yellow, red color, and red color plastid, which is carotenoid. For example, flowers. Third is leucoplast. It's a colorless plastid which is no pigment, usually found in root and stem, stars, and star storage body. But udah hanya the most important of is chloroplast. Chloroplast is site of photosynthesis in green plant and green algae. The size is 0.5 until 2 micrometer wide and 10 micrometer long. The shape is vary. The location is do not stay in fixed position. The structure of chloroplast is surrounded by outer and inner membrane. Second is extensive internal membrane system made up from extensive internal membrane system made up from vesicle known as thylakoid. Third is the thylakoid vesicle that are flattened to form discs which are stacked to form grana, which is single disc granum. Can you describe about endoplasmic reticulum?
Okay, endoplasmic reticulum, which is ER, just structure in eukaryotic cell, made up from phospholipid bilayer, a system of sacs which are enclosed membranes known as cisternae. Okay, connected to nuclear membranes, the Golgi apparatus and lysosomes, and classified based on the presence or absence of ribosome. That is rough ER and smooth ER. Okay, smooth ER, tubular or sac shape, smaller than rough ER. Okay, the function is involved in synthesis and transportation of phospholipid steroid and fatty acids. The detoxification of hazardous chemical, which is pesticide and carcinogen. Carcinogen. Okay, for rough ER, stack recycle with ribosome on their surface. Okay, the function is temporary storage for protein synthesized by ribosomes and transporting protein from one part to other part in cell using cisternal space.
Can you different between smooth endoplasmic reticulum and round endoplasmic reticulum?
Okay, for the smooth ER is synthesize lipid and steroid, metabolize carbohydrate, detoxifies poison, store calcium, glycogen, degradation site. Okay, for rough ER is has bound ribosome, secret glycoproteins, distribute, transport, recycle proteins surrounded by membranes. Okay, and last is membrane is factory for the cell. Okay, rough reticulum RR. Ribosomes are attached to the surface of SR, that is smooth reticulum, used for synthesizing protein. Okay, for the smooth reticulum SR, ribosomes are not present, used for secreting proteins. Okay, can you describe about ribosome?
Okay, ribosomes are occur in all cells, especially the actively synthesizing protein. It consists of two subunits, small and large subunit. It also synthesized by nucleolus. The location is freely floating in cytoplasm, which is free ribosome, and attached to raw endoplasmic reticulum. It's made from RNA and protein. The function is for protein synthesis.
Okay, can you describe about the Golgi apparatus?
Okay, the Golgi apparatus is occur in all animal and plant cell. It consists of a group of membrane surrounded sac. It occurs mostly in secreting cell. The function is for processing, modify, and packing new protein from endoplasmic reticulum into glycoprotein, which is protein which have carbohydrate groups. Second is secretion of glycoprotein, and third is formation of membrane and cell wall. Can you describe about lysosome?
Okay, lysosome only existing in animal cells. Spherical shape, contain digestion enzyme. That is 40 type of it. Okay, and from the Golgi apparatus. Okay, the function of lysosome is intracellular digestion via phagocytosis. Okay, destroying bacteria and foreign substance in cell involved in autolysis. Lysosome membranes will dissolve and all the digestion enzyme will destroy the whole cell. For example, tadpole to the mature frog. Okay, can you describe about vacuole?
Okay. Uh, in vacuum, it vesicle filled with fluid which is enclosed in single layer membrane known as tonoplast. Vacuum is a subfield with H2O, organic material which is glucose and amino acid. Mineral salt dissolve oxygen and carbon dioxide. In animal cell, it's a small vacuole or have none of it. Plant cells have large vacuole and increasing in size as the cell become more mature. The function is storage for waste material and cell enlargement. Water fills up with further increase the cell size. What is mitosis?
Okay, mitosis is a type of cell division which involves the division of the nucleus to produce two daughter cells. Daughter cells contain same number and same kind of chromosomes as the parent, where it is all somatic cell, which is all body cell except gametes. Okay, somatic cell is contain two set of chromosomes, one set from female parent and another from the male parent. Two set of chromosome, that is diploid cell, somatic cell. Cell with single set of chromosome, haploid cells, that is gametes. Okay, interphase divide into three subphases. First is G1 phase at first of gap. Cells grow by producing protein and cytoplasmic organelles. Cell grow and differentiation occur. For S phase, that synthesis of DNA occur and DNA replication. For G2 phase, second gaps, cell grows by producing protein and cytoplasmic organelles. And cell grow and differentiation occur. In interphase, chromosomes appear as thread-like structure, that is chromatin. In general, interphase phase where the cells prepare for cell division.
For M phase, once interphase completes, M phase begin in which mitosis and cytokinesis occur. Shortest part of the cell cycle because interphase make up about 90% of the cell cycle. Stage of mitosis. There are four phases. First is prophase, second is metaphase, third is anaphase, and fourth is telophase.
Okay, prophase, that is centrioles move apart to the opposite poles. The chromosomes curl up and condense and shorten. Okay, for late prophase, each chromosomes appear as two identical chromatids, sister chromatids attached at centromere. Nuclear membranes breaks down and then nucleolus disappear and formation of the spindle fiber.
For metaphase, centromeres attach to spindle fibers. Chromosome move to cell equator. Anaphase, centromere of each sister chromatid divide into two. Each will separate and move to opposite. In anaphase, centromere of each sister chromatid divide into two. Each will separate and move to opposite cell. Spindle fiber pull the centromere toward each pole. When the chromatid reach the pole, they will become independent chromosome.
Okay, telophase. Two set of chromosome at each pole. Okay. After that, chromosome decondense. That is thread-like structure. Okay, nuclear membranes begin to form around both sets of chromosome, and spindle fiber disappear, and nucleolus reappear. The result of mitosis is two daughter nuclei, each with same chromosome number as parent cell.
Cytokinesis usually occur between late anaphase and end of telophase. Two mechanisms. First is cell plate formation, which is in plant, and cleavage in animals. What is meiosis?
Meiosis is a type of cell division that occur in reproductive organ to produce the daughter cell, and known as gametes. Each gamete has half the chromosome number of the parent cell, that is haploid cell. Okay, genetic material in gametes different from the parent cell and each other. Where is it? In reproductive cell. Okay, the process of meiosis is involved in two nuclear division, that is meiosis 1 and meiosis 2. Each contain four phases. First is prophase 1 and 2. Second is metaphase 1 and 2. And third one is anaphase 1 and 2. And fourth one is telophase 1 and 2. Okay, the meiosis 1 is the homologous chromosome are separate into two haploid together. Haploid daughter cell, that to end to the end. Okay, the meiosis 2, sister chromatids of each daughter separate, and that which is same as mitosis. Okay, interphase, the thread-like chromosome and chromosome replicate.
For prophase 1, centriole move to opposite pole. Chromosome thicken and condense. Each chromosome become visible of two sister chromatid attached of at centromere. Homologous chromosome pair together, which is called synapsis. Each pair chromosome is called a bivalent chromatid. In each pair, one maternal chromosome and another is paternal chromosome. Chromatid crisscross along their length, which is called chiasmata or chiasma. Nuclear fibers and nucleolus are disappear. Spindle fiber is formed.
Okay, for metaphase 1, pair homologous chromosome line up at the cell daughter equator. That equator. Okay, chromatids of each chromosome are attached to spindle fiber. Okay, for the anaphase one, the pair of homologous chromosome separate from one another and move to opposite poles pulled by spindle fiber. Okay, the sister of chromatids remain attached. Okay, for telophase one, okay, chromosome reach the pole. Okay, each pole has a haploid, and chromosome sets. Okay, and spindle fiber disappear. Nuclear membranes and nucleolus reappear. Okay, cytokinin occur. Okay, for prophase 2, central duplicate and move to opposite poles. Okay, each chromosome consists of two sister chromatids that is attached to centromere. Okay, nuclear membrane are nucleolus disappear. Spindle fiber formation.
For metaphase 2, chromosome line up at the cell equator. Okay. For anaphase 2, sister chromatid separate to become independent chromosome. Spindle fibers pull chromatid to opposite pole. For telophase 2, chromatid reach pole and become new chromosome. Nuclear membrane and nucleolus reappear. Then thread-like chromosome. Cytokinesis occur and four haploid cell form. The significance is mitosis produce new cell for growth. Second is repair and replace cell that are dead or damaged. Third is asexual reproduction. For example, amoeba species. Fourth is ensure that the offspring cells are genetically identical to the parent. And fifth is preserve the diploid number of chromosomes. The significance for meiosis is allow trait inheritance in offspring or progeny daughter cell. Genes from both parents present in zygote. Then it maintain diploid number in each generation. Haploid sperm plus haploid ovum become diploid zygote. Then ensure production of haploid gamete in sexual reproduction, and after that produce genetic variation among offspring, for example crossing over.
Okay, for comparing mitosis and meiosis, is meiosis process division of cells. Both involve division of nucleus and division of cytoplasms. Okay, chromosome only replicate once in both of process.